So let's talk about the plasma membrane. What is it? Well, it is a barrier that separates the inside of the cell from the outside of the cell. It acts as an active barrier, separating intracellular fluids from extracellular fluids. Extracellular fluids are substances found outside of cells. Intracellular fluids are substances found inside of cells, like cytosol, mixed in with those organelles we talked about before. And the plasma membrane is able to play a dynamic role in cellular activities. Its structure allows it to control what enters and leaves the cell. Another name for the plasma membrane is the cell membrane. So let's look first at those extracellular materials. What are some examples? Well, interstitial fluid, kind of a fluid that cells are bathed in. Blood plasma, which is mostly water, and that's the fluid of the blood. And cerebrospinal fluid, both within and surrounding other nervous system organs, like the brain and spinal cord. Other cellular secretions like saliva and mucus and sweat also count as extracellular material because they're secreted from the cell to the outside, or extracellular space. Probably one of the most important parts of this extracellular space is the extracellular matrix. That is because this substance acts as a glue to hold cells together. It provides both cell structure and helps dictate some of their function. But let's look at the plasma membrane and look at what is it made up of. What is the barrier itself made up of? Well, it consists of many membrane lipids, and it forms a flexible lipid bilayer. What forms most of the lipid bilayer is a type of lipid called a phospholipid, but that's not all there is. There are also membrane proteins that float throughout this fluid membrane, so you could say they're integrated or integral to the plasma membrane. But also, this plasma membrane is not a static, kind of solid structure. It's kind of moving. It floats. Things can move about within it. We actually consider the plasma membrane a fluid mosaic model. It's made up of many pieces. It makes up this kind of expected pattern, but that pattern is slightly shifting and changing. There's also things attached to the outside of this membrane and on the inside. On the outside, there are surface receptors and surface sugars. The surface sugars form the glycocalyx, which you can think of as a sugar coating for cells. Many of these glycocalyx molecules allow for the cell to be recognized by your immune system. There's also other membrane structures that help hold cells together through cell junctions, like tight junctions, which hold epithelial cells together and prevent water from moving through epithelial sheets. So all of these structures attached to the outside of the cell are also making up what we call the extracellular matrix. So here's your first look at that fluid mosaic model, or at the dynamism of the cell membrane, again, which is also known as the plasma membrane. It's actually exceedingly thin. The double layer is phospholipids, but we can see all the purple integral proteins embedded throughout. There's also cholesterol, which can help with the fluidity or stability of the membrane. There's glycolipids, and there's carbohydrates attached to the outside. These are some of the glycocalyx or sugar coating molecules. And other substances like certain types of proteins and fibers attach to the cell to keep it in place and to provide more structure. And those are parts of the extracellular matrix. So let's continue talking about that lipid bilayer. It is 2 layers of phospholipids, and we have proteins dispersed throughout. Again, most of that bilayer is phospholipids, but we can't overlook cholesterol and the glycolipids. The glycolipids are part of the sugar coating, and they tend to be found on the outside, and they're called glycolipids because it's a lipid bound to a sugar group. The phospholipids, while primarily being kind of the star of this lipid bilayer, are also going to work with cholesterol to help stabilize the fluidity of the membrane. Depending on how much cholesterol is in the membrane, you may have a more or less fluid membrane. But we don't want to just kind of overlook the phospholipids. Again, they're a major part of the plasma membrane. Phospholipids have a polar head and a nonpolar tail. In the head is a phosphate group that is charged, which accounts for the hydrophilic or water-loving nature of the phospholipid head. The hydrophobic tail, however, is composed of 2 fatty acids, which are chains of hydrocarbon, and that is why they don't like to be near watery environments or are hydrophobic. Here we can see a picture of multiple phospholipids arranged with their heads facing outward and inward, both towards the extracellular space and towards the inside of the cell or intracellular space. Their tails are actually sequestered away from these watery environments, and that's because they're hydrophobic. So this is the general arrangement of the phospholipid bilayer. What's missing in this basic image is all the other structures inside and outside the cell to provide support to this cell membrane and the integral proteins that would be embedded within. So now that we've looked at the membrane lipids, we can see that about 75% of that lipid bilayer is phospholipids. 5% is glycolipids, and 20% is cholesterol. And remember, cholesterol can help increase or decrease membrane fluidity. When it increases membrane stability, it's really decreasing fluidity and kind of making our fluid mosaic model a little more stagnant. But now that we've talked about the basic structure for the plasma membrane, let's talk more about its function. We know it's a mechanical barrier, but it's also selectively permeable. That means that not everything gets through. It might also depend on the electrochemical gradient, or kind of the charge of things across the membrane, on whether or not they get through. That's actually something we look at for muscle and neuron function. The plasma membrane also helps with communication and cell signaling. So before we go further, we're going to talk more about the membrane proteins. And as we build and talk more about everything in the cell membrane, besides just the phospholipids, some of those functions we just talked about, like cell signaling and communication, will be elucidated or make more sense. So the first type of protein we're going to look at is an integral protein. This is one that is integrated or firmly inserted into the membrane. So many go across the membrane, so we consider them transmembrane proteins. To go across the membrane, they have to have both hydrophobic and hydrophilic regions because those regions exist within the plasma membrane. Some of the most common types are transport proteins, or ones that carry out enzymatic activity, or are receptors for signal transduction, like that of using amino acid hormones. There's also peripheral proteins. These do not go through the membrane. Instead, they attach loosely to integral proteins. They also can have enzymatic activity and provide support, but oftentimes they're motor proteins or provide cell-to-cell links. Many of the cell junctions, like holding cells together or attaching cells to other structures, are provided by peripheral proteins. They exist both inside and outside the cell. And some cells don't form junctions. Some cells are free. They're not bound to any other cell, like spermatozoa or sperm cells and blood cells. But many cells are bound together, and they form tissues and organs. Some of the most important membrane junctions are of three types: tight junctions, desmosomes, and gap junctions. Tight junctions are impermeable. They are specialized junctions that use studded proteins to hold two cell membranes together. They hold them together so tightly that even water can't get through the membrane where these tight junctions exist. Epithelial cells generally have quite a few tight junctions, and epithelial cells oftentimes therefore show up in sheets of cells. Then there's the desmosomes. Desmosomes are more like molecular zippers. They use these specialized plaques located on the inside of cell membranes and protruding types of linker glycoproteins called cadherin to link cell membranes together. But these are not impermeable. Things can get through, at least through in between the cells, not through the plasma membrane. Instead, what the desmosomes are for is more of an anchor. They're thought of really as an anchoring zipper. So they're really a molecular zipper that provides structure and support to two cells near each other and allows some stretch and give. That stretch and give is really important when it comes to the function of desmosomes, because desmosomes reduce the chances of tearing when subjected to pulling forces, and that's important in the skin and heart muscle. Without desmosomes, on top of having tight junctions in your skin, whenever you accidentally kind of trip and bump into something, your skin would shear off because no stretch would be possible without desmosomes. So thankfully we have desmosomes, and at most our pride might be hurt, and we might have a little bit of a bruise. But our skin oftentimes doesn't just shear off because we bump into something. And our heart muscles constantly contract with a lot of force. If they didn't use desmosomes, they would likely tear apart upon contraction, and your heart would barely beat more than once. And then last but not least, we have our communication junction, called a gap junction. They're called a gap junction because there's a gap in between the proteins that make a tunnel between one cell to another. Small things can go through this gap like small ions, but something like a really large carbohydrate is not getting through this gap. So let's review. Tight junctions are integral proteins where we see molecules between two separate cell membranes fuse, making an impermeable junction. Desmosomes are molecular zippers that provide anchor and stretch. And gap junctions are our communicating junctions that allow really small things to pass through from one cell to another. Gap junctions are really common among muscle cells, especially those that need to communicate cell to cell very quickly. All right, next we're going to be looking at membrane transport. And so now that we've reviewed the plasma membrane, we can talk more about that selective permeability and the other functions of the membrane and how things move in and out. So that's what we're going to look at next.